Basically, there is three main subtopic to covered, which is 4.1 Mendelian genetics, 4.2 deviations from the Mendelian inheritance and 4.3 genetic mapping. There are learning outcome related to the chapter, which is for 4.1, we need to define the terminologies used in genetic inheritance, state the characteristics of Mendels Pea Plants, state Mendels' first law, state Mendels' second law and construct genetic diagram. For 4.2, we need to define types of inheritance that deviate from Mendelian and 4.3, we need to define genetic mapping. Okay. Let's start with 11 important terminologies that used in genetic inheritance. Okay. The first term is allele. Allele is alternative forms of a gene for a trait occupying the same locus on homologous chromosomes. In this diagram, there are two types of alleles, which is allele for purple flower and allele for white color, in the same locus on homologous chromosome. Genes. Genes is a heritable unit consisting of a specific nucleotide sequence in DNA that controls a specific characteristic or phenotype of an organism. Locus. The position on a homologous chromosome where a gene is located. Genotype. Genotype is genetic composition of an individual, combination of alleles it possesses, or the alleles possesses by an individual. In this diagram, their type of genotype, which is capital B that indicate brown color eye, capital B and small b that also indicate for brown color eyes, and small b that indicate for blue color eyes. Phenotype. Phenotype is the observable characteristics of an organism. For example, blue eyes or brown eyes. Homozygous. Homozygous is an organism that has a pair of identical alleles for a gene. For example, either homozygous dominant capital BB or homozygous recessive small bb. Heterozygous. Heterozygous is an organism that has two different alleles for a gene, which is capital B and small b. Dominant allele. Dominant allele is an allele which expresses its phenotype even in heterozygous form, while recessive allele is an allele that only expresses its phenotype when present in the homozygous form. In genetic inheritance, there are two types of cross, which is test cross and self-cross. Test cross is individual of unknown genotype with dominant phenotype is crossed to a recessive homozygous individual. If one of the parent is homozygous dominant, capital P, all the possible offspring should be all dominant phenotype, which is purple flower. If one the parent is heterozygous, capital P small p, the possible phenotype is 50% purple flower and 50% white flower. Self-cross or self-pollination. Self-cross is the transfer of the pollen grain from the anther to the stigma of either the same or genetically similar flower.
One of the founding fathers of the USA, Benjamin Franklin, profoundly said, "By failing to prepare, you are preparing to fail." And whilst I'm pretty sure he did not say this with biology in mind, we are going to hold him to his word and prepare ourselves by making sure we know what these key words all mean. Gametes are reproductive cells, or sex cells, so female gametes are the ova or egg cells, and the male gametes are the sperm cells. DNA is tightly coiled around proteins and is packaged into structures called chromosomes. They are found in the nucleus of cells. A gene is a unit of hereditary material. Each chromosome contains thousands of genes made up of DNA. The gene codes for a particular characteristic of the organisms, such as eye color or blood group. An allele is an alternative name for a gene, representing the different versions of that gene. So whilst the chromosome from your mother and father both have genes for hair color, body height, and lipoma formation, they may have different alleles. So your mum may have a red hair allele, whereas your dad has a brown hair allele. The phenotype is the outward appearance or characteristics of the organism depending upon genes. So the phenotype for the red hair gene is actually having red hair. The genotype is the set of genes in our DNA which are responsible for the phenotype. Alternative forms of a given gene are called alleles. When an individual has two of the same allele, so both coding for red hair, they are homozygous. Both genes are identical. Whereas if one allele codes for red hair and the other for brown hair, they are heterozygous. Two different forms of the same gene are present. When two different alleles are present, so are heterozygous, one allele will be dominant. This allele will influence the phenotype. So brown hair is dominant over red hair, so the offspring is more likely to have brown hair. So in this example, the red hair allele is recessive. This means the gene is not expressed in the phenotype. Recessive genes are only expressed when the other gene is also the recessive form, so it needs to be homozygous, both carrying the red hair allele. And finally, what do haploid and diploid mean? Haploid cells have one set of chromosomes and is labeled as an N. Diploid cells have two sets of chromosomes, 2N. Most cells are diploid, so in humans we have 23 pairs of chromosomes, one from our mother and one from our father. All of our cells are diploid other than the gametes, so the egg and sperm, which are haploid. When the sperm fertilizes the egg, the chromosomes pair up and become diploid. So they have to start at haploid so that there is room to pair.
Gregory Mendel was an Austrian scientist. He is known as the father of modern genetics. To understand more about Mendels experiment of pea plants, please watch the video.
How is variation inherited? Let's see how the variation takes place and how the variation is inherited from one generation to another. The human population has two variants of earlobes. Some have free earlobes, while others have attached earlobes. A particular mechanism is responsible for such variations. The inheritance of traits is related to the fact that both the father and the mother contribute equal amounts of genetic material to the child. If we try to correlate the earlobe of a child with that of the parents, we understand that the father's gene for earlobe is dominant, because of which the child developed an attached earlobe. The process of inheritance can be easily understood through Mendel's experiments. Let's know more about Mendel and his work on pea plants. Did you know that Mendel was educated in a monastery and studied science and mathematics at the University of Vienna? He wanted to be a teacher, but he failed the teaching examination. However, he did not lose hope. He returned to the monastery and started growing pea plants in the garden. Mendel blended his knowledge of science and mathematics to keep count of the pea plants exhibiting a particular feature in each generation. Mendel researched heredity and arrived at the laws of inheritance. He used a number of pea plants with various traits such as tall or short plants, round or wrinkled seeds, white or violet flowers, and so on. The process of inheritance can be easily understood through Mendel's experiments. Let's first understand the Mendel's experiment, in which inheritance of only one character is used. For example, the height of the plant. Such an inheritance pattern in which only one trait is used is referred to as monohybrid inheritance. Mendel used pure breeding tall plants with the phenotype TT and pure breeding dwarf plants with the genotype TT to reproduce by cross-pollination. Let's draw a Punnett square to find the inheritance. The Punnett square shows the gametes produced by the parental plants, uppercase T and lowercase t. And the F1 generation has all uppercase T and lowercase t progeny. In first generation or F1 progeny, all of the plants were tall. This means that only one of the parental traits were visible. The trait which is visible in F1 generation is called the dominant trait, and one which remains suppressed is known as the recessive trait. Here, tallness is the dominant trait and shortness is the recessive trait. All the F1 plants were tall, TT, as that of the parent tall plant phenotypically. Mendel then allowed the F1 tall plants of the genotype uppercase T and lowercase t to reproduce by self-pollination. As a result of random fertilization, the second generation or F2 progeny were not all tall. Instead, 1/4 of them were short. This indicates that both the tallness and shortness traits were inherited in the F2 plants. Now let's see the entire experiment in the form of genotypic and phenotypic representation. It is seen that random fertilizations result in 1/4 uppercase TT, half uppercase T and lowercase t, and 1/4 lowercase tt. This gave the genotypic ratio of 1:2:1. 1 pure tall plant, 2 hybrid tall plants, and 1 dwarf plant. Thus, 75% of the progeny produced in F2 generation were tall, and 25% were dwarf. This gave the phenotypic ratio of 3:1. What happens when pea plants of two different characteristics are bred with each other? Let's consider a hypothetical situation. We cross a plant producing round and yellow seeds with a plant producing wrinkled green seeds. The inheritance of two such characters is known as dihybrid inheritance. The entire F1 progeny produces round and yellow seeds. In such a case, round and yellow seeds would be the dominant traits. The next step is to self-pollinate the F1 generation. Mendel found that some plants of the F2 progeny were similar to their parents and produced round yellow seeds, and some of them produced wrinkled green seeds. However, some plants of the F2 progeny even showed new combinations like round, green, and wrinkled yellow seeds. Therefore, it can be safely concluded that the round or wrinkled trait and the yellow or green colored traits are independently inherited, and they are known as recombinants. How do the traits get expressed as characteristics? Cellular DNA is the information source for making proteins in the cell. The proteins control the characteristics in the plant. Well, to make it clear, consider a plant with tallness as a characteristic. We know that plants have hormones that trigger the growth. Assume that an enzyme performs this process. If this enzyme works efficiently, a large quantity of hormone will be made, and the plant will be tall. However, if the gene for that enzyme has an alteration that makes the enzyme less efficient, the amount of hormone will be less, and the plant will be short. In this module, you learned that the inheritance of only one character is referred as monohybrid inheritance. The inheritance of two characters is referred as dihybrid inheritance.
Why Mendel used pea plants in his experiment? There are a few characteristics of Mendel's pea plants, which is: pea plants can self-pollinate or cross-pollinate, produce large number of offspring from each mating, short life cycle or mature quickly, many easily observable characteristics, and easy to grow. This is the example phenotype of Mendel's pea plants. For flower color, the dominant phenotype is purple color while the recessive phenotype is white color. For plant height, the dominant phenotype is tall while the recessive phenotype is short. For seed color, the dominant phenotype is yellow and the recessive phenotype is green. For seed shape, the dominant phenotype is round while the recessive phenotype is wrinkled. For pod color, the dominant phenotype is green while the recessive phenotype is yellow. For pod shape, the dominant phenotype is inflated, while the recessive phenotype is constricted. And last, for flower position, the dominant phenotype is axial while the recessive phenotype is terminal. Mendel's first law, or law of segregation. Mendel's first law states that the two alleles for a heritable character segregate during gamete formation and end up in different gametes. Mendel's second law, or law of independent assortment. Mendel's second law states that each pair of alleles segregates independently of each other pair of alleles during gametes formation.
After his experiments of monohybrid and dihybrid crosses, Mendel concluded that these are certain factors which are passed from one generation to the other. But he explained this better with the help of his first law in genetics. The first law in genetics is called the law of segregation. To understand this law, we first need to understand the concept of alleles. We know that genes, in simple terms, are the units of heredity that carry genetic information. Alleles are nothing but the different forms of genes. Let's take a simple example to understand this. Assume we have these two chromosomes. Here we have genes for the character height. Can you tell me which genes could possibly be present here? we can have either both capital TT or both lowercase tt, and we can even have one capital and one lowercase tt as the gene set. Now here, the individual T, that is the single gene, is nothing but the allele. So the alternative form, that is either dominant or recessive, is called allele. In simple words, one single T or T is an allele. Now that we know what alleles are, let's try to understand the law. There are different definitions you will find for this law, but let's pick the simplest one. It says that during gamete formation, the alleles for each gene segregate from each other such that each gamete formed carries only one allele for each gene. It may still sound a bit confusing. To understand this, let's take a simple example. Let's assume we have this single cell with only 2 chromosomes in it. The chromosomes have a set of genes CC for the skin color in an individual. Now to understand the law, let's break it into parts. The first part says, during gamete formation. This is the process of meiosis. It is the type of cell division which gives us 4 cells with half the number of chromosomes. The second part says, the alleles of each gene. So here, C and C is what we are referring to. That means one allele from the gene set. Now what's the next part? The next part of the statement is segregate from each other. This means the alleles will separate. They will not remain together in the process. And now comes the last part. It says that one gamete carries only one allele for each gene. Now this part means that at the end of the meiosis process, we will get 4 cells such that each cell will have only one allele in it. So here, 2 cells will have capital C and the other 2 cells will have the lowercase c allele. Thus, each gamete gets only one allele. To summarize, we can say that the alleles for a gene segregate during gamete formation. To be honest, this law sounds very obvious to us now. It's like having a big book with many chapters in it, and then we make smaller books for each chapter. It is obvious that the smaller books will have the content resembling the content of the big book. This law sounds obvious also because we know the process of meiosis. But nobody knew this back in the earlier times. Hence, it was hypothesized by Mendel, and then it was accepted as a law later. So what's the next law? It's the law of independent assortment. Does this mean that the genes assort independently? Or is there anything else that the law specifies? Let's find that out in the next part. There is one common thing among sea waves, dust particles, and a dice roll on the floor. Any guesses what that common thing could be? It's randomness. Randomness is commonly found in nature and in several things around us. But can you state an example of randomness that occurs inside us? Though our body seems to be organized, there are a few processes that have no fixed pattern. One of the processes is that of the alignment of chromosomes on the metaphase plate. Let me elaborate. Imagine this to be a cell that's about to undergo division. For now, let's assume it's undergoing meiosis. We know that during cell division, a stage named metaphase is reached where the chromosomes align themselves on the equatorial plane. In other words, on the metaphase plate. So can you tell me how these chromosomes will align before they line up on the plate? Will all the red chromosomes from each pair line up on one side, or will they align in an alternate manner? Or maybe like this? Or will the alignment be like this? There are numerous ways in which this can happen. And this is random. There is no fixed pattern or a principle of sequence that the chromosomes follow when they align. Now, if the chromosomes get separated randomly, it's obvious that the genes present on them will also get separated randomly. So let's say this pair has the genes capital A and lowercase a. This pair has the gene set capital B and lowercase b, while this one has capital C and lowercase c on it. We are considering all the sets to be heterozygous in order to understand the concept better. So when the chromosomes get separated in the respective cells, it's obvious that the genes will assort independent of each other. There is no thumb rule that all the dominant alleles will perpetuate in one cell and all the recessive alleles in the other. They can assort independent of each other. This is exactly what Mendel had to say in his second law of genetics. It's named the law of independent assortment. The law says that genes for the different traits assort independent of each other during gamete formation. Let's understand this with the help of this cell. The first part says genes for different traits. This means we are addressing the genes which represent diverse traits. So here we have the gene sets AA, BB and CC on these respective chromosomes. These are the genes for different traits. Now the second part states assort independent of each other. This means the genes will not have a fixed pattern to follow when they segregate in the cells. So here, the alleles of the gene set AA will assort independent of the other two gene sets. It's not mandatory that all the dominant alleles will get assorted in the cells together. So the allele capital A can get assorted with any allele from the gene set BB and CC. And that's the reason why we get the gametes with different possible combinations at the end. To quickly review, the assortment of alleles is purely random and does not have a predictable and fixed pattern during the process of gamete formation. That is what the law says. There can be more possibilities of obtaining different combinations depending upon how the chromosomes align themselves on the metaphase plate. Now tell me, which type of a cross will help us understand this law better? A monohybrid cross or a dihybrid cross? Think about it. Let me help you with this. If it's a monohybrid cross, then there will be no independent assortment of the different alleles. And why is that so? That's because we are considering only one character at a time. So we will be dealing with a single gene set. And thus, this law is best understood when there are more than one character studied at a time. This is nothing but the dihybrid cross or higher crosses which involve more characters.
You will learn the next subtopics. 4.2 Deviations from Mendelian Inheritance and 4.3 Genetic Mapping. In the next video, please scan the QR code on the next slide as the enrichment to this lessons. Have fun!